All right, it's 4:01. Welcome everybody to our second Tuesday seminar. Today's seminar will be talking about how to read an academic paper. We have two special guests with us from Colors of the Brain, but before I get to that, I wanted to send out a couple reminders just for all of you. Number one, for the students that are in the GRE, there is, I believe, an office hour later this week. If you have questions about that, please reach out to your program coordinator. And then also the Summer Research Conference registration for all of the presenters will be July 11th. So please don't forget about that. And your program coordinator should have sent you some information about that. And if you haven't gotten it yet, certainly reach out to them. Other than that, I think we'll get started with introducing our speakers. We have two grad students, Pamela Riviere and Michael Preston, who are here on behalf of an organization called Colors of the Brain that started a few years ago. Colors of the Brain has a mission to provide resources for students that are underrepresented in brain and psychological sciences. And so they definitely want to have more of those students represented. And so they are coming here. They are currently grad students. Pam used to be at Boston University as a neuroscience major and is now in the Cog Sci pursuing her PhD. Michael came from American University in neurosciences and is now pursuing a neuroscience PhD here at UC San Diego. And so with that, I'd like to turn the table over to them so that they can get started with the presentation. Thanks.
Thank you so much. So yeah, today we've been invited to do a little tutorial or workshop on how to read an academic paper. And so let's see. Cool. So the first question you might want to ask is, why would you want to read an academic paper? So first of all, a lot of you might be joining or have started working in new labs, and you're beginning to get involved in an ongoing research project. And so in this case, you might want to get oriented to what the research project is about. And you'll see that there's a central scientific question that your research team is asking. And so you can read an academic paper to get oriented to what is currently known about some research topics. So what do we know? And also, what do we not know yet? So a lot of research is just identifying gaps in our knowledge and trying to plug those gaps with like a little bit of like insight that we generate from a given study. And so in order to understand the contribution that your research team is making into plugging this gap, you kind of want to know what you know and what you don't know. Additionally, whenever you're beginning to think about your own projects that you might want to design yourself, you might want to read academic papers so that you get inspiration. So there's something really exciting about seeing what different research groups have already accomplished and how they have approached interesting questions in the past. So you can also follow suit and generate research questions and research ideas, and also the methods for how to go about those. Okay, so now we know why we want to read a research paper, but what is it? So what is the structure of an academic paper? What is it about? How is it set up? So you will encounter a lot of review papers out there. So review papers are a little different than what are called primary research articles, and the rest of this talk will focus mainly on these primary research articles. But when we're thinking about review papers, we're thinking about getting oriented, getting a broad summary of what the field already knows about a given topic. In this example review, you know, like the title is pretty broad. The reviewer or the author is trying to ask a question about what we know about this particular topic. So in this case, it's just like, how do neurons control musculature? So how do neuronal activity translate into muscle control? And so what they're going to do is cite or make reference to a lot of different primary research articles. And so you can already tell probably from these titles, so like millisecond scale motor encoding in a vocal area, or variability of motor neuron spike timing in the muscle of aplysia, whatever that is, or force encoding in muscle spindles. So all of these primary research article titles are way more specific than the title of the review paper that they're being cited in. And so this structure of having like a main idea and a main question that is being covered in the research paper and also having the backbone of evidence that is provided by individual contributions in these primary research articles is one of the ways in which you'll be able to get oriented to a new field. So anytime you have no idea what kind of primary research articles to look up in order to like know about some broad topic, look up review papers. So you can just type in reviews on subject X, and a ton of things will come up that will give you pointers to looking into primary research articles that might be helpful for getting oriented into this literature. So the rest of this talk will focus on primary research articles because they're super dense and very technical, and they're often really hard to read. And so that's what this tutorial will be about. So take it away, Michael.
All right, so how do you go about finding these primary research articles and reviews? Here I've included a list of databases that you might access. I've launched a quick poll just to see what sort of resources you all are using currently. If you don't mind taking a second to fill that out. The first resource on this list is just a list of databases. This could be all the places you might look. This is provided by the UCSD library. I see you guys filling it up here. Our next database is Web of Science. This is a more general database here, which I think applies to many of you. Those in biomedical sciences specifically, I think you'll probably rely on PubMed. The different pros and cons of these different search engines are explored in greater detail on this first resource link here provided by the library. And Google Scholar is also a great resource. This differs slightly from these other databases in terms of how specific you can get with your search, these different search terms, and also whether you're searching through just the abstract or the full article. So depending on your goals, you might utilize one of these different resources. So looking at this poll here, it looks like 81% of people have checked Google Scholar, with about 50% are using PubMed and JSTOR, and around 20 using Web of Science. So that's pretty interesting. Thank you. A few other resources I've shared here include this PubMed online trainer. The National Library of Medicine provides some great tutorials. These are videos on how to get, again, conduct these more advanced searches to really hone in on what you're looking for. This Boolean operator guide will also help you in conducting more specific searches. And I'll also include a link here to this literature scanner. Those of you with some coding experience, you can actually explore these databases from kind of a higher level to a meta-analysis of what papers are out there. So that might be of interest to some of you. These are a couple supplementary resources that might help you to find connected papers. Let's say you have a paper of interest and you want to find some similar papers to that. Connected Papers is a great resource. I've also included Citation Gecko. Here's if you have multiple papers and you want to find more recent works that cite both of them, or more foundational work that is cited by 2 or even more papers. Both these resources will help give you a good sense of the field and what knowledge is already out there. On this next slide, I've just included a quick walkthrough of just this connected resource website. Let's say I have a paper on scale-free brain activity. I can quickly pull that up either by the title or the DOI or even a URL on a database like PubMed. And you get this interactive graphic where you can see similarities of papers, citations, the year they were published in this compact format. You can click on anything in this web and pull up some basic information. There's also these additional functionalities to find prior works and derivative works. Again, here you can find papers that you can basically see this evolution of thought, see how papers have influenced each other and our understanding of these topics have evolved over time. There's also some resources to interface with the publishing sites directly, or even a lot of times just pull up the PDF. So I really recommend checking out these supplementary resources in addition to these classical databases and search engines.
Cool. So, and sorry, MJ, go ahead.
Yeah, I was just gonna say, now that you've found your research article, how do you actually go about reading it? This next section, we're gonna break down into these different components you might find in an academic paper. I have them outlined on this next slide here. We're gonna be walking you through the abstract, the intro, methods, results, conclusions or discussion, and also the reference section. So again, is the abstract. In its most basic definition, your abstract is going to be a summary of your article. Basically, the goal of your abstract is to help the reader decide whether or not they're going to read the full research article. So oftentimes, the abstract will include all these major components that I just mentioned of the full manuscript, and that way it's kind of a mini version of the paper. And also, I want to note that visual abstracts are becoming more popular in a lot of journals, especially with social media. Visual abstracts can help you get more clicks, more attention to your paper, help you communicate your science to a larger audience. Yeah, so that's another thing to keep in mind here. On this next slide, I have an example of an abstract and kind of just a protocol I like to go through if I'm reading an abstract or even writing an abstract. It's actually break it down into these components, and you can get a better sense of what information you might find in the article. So ignoring the details here, you can just see how we highlighted these different sections. They're first introducing what knowledge is known, then they're introducing the methods or what these authors did, and then really the bulk of the abstract is going to be the results. When this is a primary research article, these might be empirical results. If this is a review or another academic paper, these might be main arguments. But this will usually be the bulk of your abstract. And then they'll typically close with some sort of a closing, how this relates to the hypothesis or maybe the interpretations of these results. The first section of the main body of the paper is your introduction. An introduction is going to introduce the relevant background information necessary to understand the article. A key thing about the introduction is that it identifies gaps in the current knowledge that will be addressed in the current paper. That's definitely something you want to look out for when you're reading an introduction. So the goals of an introduction are to contextualize the paper in the larger body of literature. So in this way, they're generally going to cite a lot of things, a lot of foundational work that led to our current understanding of a topic, or maybe explain some of the assumptions that the current paper relies on. And also, an introduction is going to explain the significance of the paper. Why does it matter? Why are these results important? So for yourself, as you're reading an abstract, some things you might want to do is identify the main hypotheses and motivations for the study. This is again going to be related to these gaps in the current knowledge and also the significance. And you can identify relevant literature for future review. So like I said before, it's going to contextualize the current work in the larger body of literature. So it might make reference to some topics or concepts you're not as familiar with. And this is really a great place to find additional resources to supplement that background knowledge. Another thing to note about the introduction is it typically takes this funnel structure where you're starting more general and you're really honing in on exactly what you're going to do in this paper. Again, ignoring the details here, this is the same article as our abstract about memory. The authors here start very broadly, just introducing the concept of memory. Then they introduce multiple existing models of memory before honing in on one specific model, then detailing the predictions of that model, and then getting to exactly those predictions that this paper will focus on. So in that way, you're starting broad and really honing in on what you're doing in this paper. All right, I'll take a minute to take any questions before passing it over to Pam. All right, thanks a lot. And feel free to put any questions in the chat throughout. We'll try to monitor it as well.
Yeah, so, and also I'm about to start my next section, which is about reading the method sections of papers with a question. And so my question is kind of weird, but I promise it'll be super relevant. So the question is, has anyone cooked a dish or meal recently, or had like a parent or a family member, some relative make a dish? And if you had anything like this, put it in the chat and let's see what it is. Like, I would love to see the array of meals that people either make or have relatives make for them. Kimchi, watermelon salad, oh, kimchi fried rice. Oh, nice. Okay, cool. Tomato tart. Oh, brownies are a classic, wonderful one. Mac and cheese, very good. Scrambled eggs, chicken rice, spinach, boba tea. Okay, cool. Ants on a tree. Okay, Thai curry, Very, very good. I had a Thai curry last night that my boyfriend's mom made for us. And so cool. All of those are fantastic, fantastic, fantastic. So, and I got some recommendations for myself just now. And so the question is here, did you or your relatives make or use a written or oral recipe, you know, like something that's been passed down in your family? And if you did use a recipe or some form of recipe that like your family uses, please say either yes or no in the chat, and I'll just see the running list of yeses or nos. Okay. Yep. Got 2 yeses so far. Yes. Very good. Yeah. So it overarchingly, oh, yes and no. So somebody said yes and no. Okay. Somebody said no. So wow, the first 10 ones were yeses, and then last ones are nos. But so that's really interesting. Often our method sections and papers are actually kind of like recipes. So that's kind of weird, but the idea is that a good recipe helps you make a dish like somebody else made it. And so any changes that you make to the recipe will end up changing the final product in maybe small or maybe very large ways. So a method section in a paper, and every primary research article has some form of method section, lets other people know how you went about answering a particular research question. And in science, as in cooking, this improves reproducibility and it helps compare across results. So it's totally cool to make a recipe at home, cooking that, you know, like, or you make a dish and you don't really use a recipe. You just like kind of generally know what you're going for. That's totally fine. But in the context of research, we really always want to be documenting exactly how we did a particular experiment so that somebody else can replicate that exact same sequence of steps and hopefully get the same result. And this helps us compare, you know, like if 2 different people run the same experiment, but they get different results, you're like, what are those results differences down to? You know, what gave rise to those differences? So let's see. So recipes are often multi-step processes that can involve making multiple parts that you later put together in your final product. So it's kind of like making a pizza, where you would first make the dough, and then you make the sauce, and then you have some instructions on how to put the pizza together. And so notably, if you change up the recipe a little bit, your pizza will look a little different than the pizza the recipe leads to. So something to always remember about methods is that variations in methodology is going to lead to a variation in results, like whatever, 90% of the time. And so now that we know why method sections are important, we still have to read them, right? And so here I have an example method section from a paper, and right away we can see that it's kind of like blocky. They do helpfully like break it up into smaller subsections of smaller sub-methods that they went through and they documented, but it's still pretty technical. So how do we read a method section in a way that is not like super tedious and boring for us? Well, the first thing to remember is that like methods, like a recipe, actually maps onto like very concrete things out in the world. So like it's not all necessarily super abstract. You can try to figure out like some verb or some noun in a method section and try to figure out what that maps onto in the lab. Or if it's a mathematical or statistical method, what does that variable map onto in the world? And so we're going to go through a very short tutorial on how I read method sections when I'm totally unfamiliar with a particular research area. So first of all, I start at the very beginning. You'll see that I blocked out, or I grayed out this like rest of the recipe or rest of the methods for myself. So I actually get really unfocused often. So I often just like try to like ignore information that I'm not currently processing. So I just did that for myself. And then I start at the very beginning and I look, and the first verb, I have no idea what it is. So I just look it up. I go to the internet and I search on, I search for photo tagging, and, like photo tagging might bring up different weird results. So I will just make sure to narrow down my search by saying neurons. So photo tagging neurons. And I can already see in the images that this verb seems to map onto something about somehow marking neurons in a way that you can identify them later, maybe. So, and maybe it has to do with something like color-based identification. And so then I look at the rest of the search results, so like the textual search results, and I see that there's a whole paper on tagging neuronal populations. And maybe I don't read the whole paper because I'm trying to read another paper. So I'm just gonna look at I'm just gonna look for surface level information that's gonna get me through the methods section. So maybe I see, oh, cool, this is the function of it, or this is the role of this verb. It's about like monitoring activity in specific groups of neurons. So photo tagging is whatever. I'll write a note in my notebook that says, okay, photo tagging is for tagging neurons to monitor their activity. And I mean, everybody has different ways of writing notes about things, but I find it helpful to write in a notebook. Other people find it helpful to write on computers. But either way, the act of writing things out or typing them out helps you consolidate it a little bit as you go through. And that's just like a tip that I have. It's just something that's helped me. So cool. Now that I've already gotten like a general sense for what photo tagging is and what it's for, I just move on. So you'll have time later to talk to your research colleagues and other people and ask them about like, so what does the setup for a photo tagging experiment look like? Like, how do you actually do this in the real world? And that's going to become important for you if you want to go into this kind of research. But otherwise, I just kind of stay, just for the paper's sake, just to get through this paper, I just stay at the surface level information for now. And then I go on to the next foreign noun that comes up. It's like, what is CHR2? And it's like, well, I just look it up and I can already see that there is some information on it being like some sort of like light responsive type of thing, light responsive molecule or protein or something. And I can see what its like role is in the study. It's like to enable the control of neuronal activity, maybe at some time scale. So if you go through this process, it's going to be super slow, but this is like the most surefire way to get to understand roughly what a given study or how a given study was performed. And so there are some principles that will apply to research disciplines across the board and maybe life in general. And the first one is to just like, if information is overwhelming, just break it up. Break it up into smaller pieces and identify what you don't understand. And then second, look up and just ask your research team about anything you don't understand. There's no shame in not knowing something. You will just like have a moment of not knowing, and then afterwards you will know something about it. And so you'll get to see your own delta, your own progress, and you'll just like be able to speak more fluently in the lab and contribute to lab meetings or journal clubs and interactions with your postdocs or principal investigators and so on. Okay, so now that we're done with the methods, we're going to apply the same set of principles of breaking up information to go through the results and the figures of a really dense technical paper. So papers often have like multiple streams of information that they try to like combine in order to provide evidence, like research evidence for some claim or some hypothesis that was made in the introduction, which Michael talked about. So in these results and figures, you'll note that there's kind of three broad categories of information source. So there's the graphics of the figure. There's also the caption that goes with the figure and explain what all the symbols in the figure mean. And then there's also the main text in the paper that goes through or describes the results from that figure. So I also have like a set of like principles to think about how to get information and get results and figures, and these keep hypotheses are that were made in the introduction article, and second, for or against these claims, hypotheses, or arguments that are made in the results and the figures. So this is how I just like approach results and figures. And again, I'm just going to break things up. So first of all, like a lot of figures have multiple sub panels to them. And so I can't deal with looking at panel A, B, C, and D all at the same time. So again, I focus in on panel A just to start. And so this is just the graphic. And I can already see tons of symbols that like, I just don't know what they are yet. And so then I'll look into the caption to try to get oriented to what panel A specifically is talking about. So again, I'll ignore everything that talks about any other panel, any other source of information. So I already know what kind of neuron they're talking about from this first like sentence in the caption, and I can look at what these initials mean, these WSs. So the caption will provide a lot of information about the symbols, the abbreviations that are all in the figure graphics. Then after I've taken a look at the actual graphics and at the caption that goes with the graphics, I'll take a look at the main text, especially if I don't feel super oriented to why they showed me a particular set of symbols. I'll just try to see if the main text has some information about why they think that this is important for me to see. And so I, again, take out any sort of information that's talking about anything other than figure A. And I can already see that the very first part of this main text has some general background information for the figure that tells me why they made the figure. And they even have a hypothesis at the very end of this paragraph that the figure is supposed to test. So this is how I generally break up these multiple kinds of information. Graphics, caption, main text. Graphics, caption, main text. And I rinse and repeat anytime I am reading a research paper that has a lot of figures in them. Okay, so next up, we'll go briefly through a different kind of paper. So the humanities and social sciences might have papers that are less figure heavy, so they're more essayistic in a way. But regardless, we'll still have very similar structure to the way that an argument is made in support of a particular hypothesis or claim. So in this particular paper from the history department at UCSD, we have similarly an introductory segment. And then if you keep reading, you'll be able to identify what the main argument the author is trying to make is. So often people will be really explicit, authors will be really explicit about what their argument or core thesis is. And so here I found this by reading this article argues for a broader understanding of X and Y. So people will tell you, authors will tell you exactly what they're trying to do with this piece of writing. And your job is to find what the evidence is that they present, and to think critically about whether you think the evidence does or doesn't support the main argument being made. These papers will also often have like description of the method. So in the case of historical archival work, you might get a sense for what the kind of data is and what the limitations on the available records are. So how sparse or spotty is your data set, and also what the inferential process is for like filling in the gaps inferentially in this data set. And then lastly, you find the evidence that is presented and you try to like figure out whether it makes sense in light of the claims being made at the introduction. Cool. So in summary, we're always going to break up information into smaller pieces. We're going to look up or ask about anything that we don't understand. We try to keep the main argument in mind as we read. And when we find evidence that is presented, we'll always ask ourselves, Does the evidence convincingly support the claim or argument? And often what is really helpful is to try to articulate it, whether writing it down or telling people about this, why you do or don't find a piece of evidence convincing. That is going to help you consolidate information and be able to then talk about it again in either your like research writings or also like talking to your colleagues about this stuff. Okay. So this is just a summary of wait. Cool. This is this is all you.
All right, so the final section you're going to encounter is the discussion and the conclusion section. Sometimes this is a single section, sometimes they're split into two. But the main focus here is going to be on interpreting the results. So in the results section, they're going to say what they found, maybe state the main arguments, but here they can refer back to the hypotheses and talk about what new things we learned and basically what are the implications of that. Some additional elements you might find in the conclusion or discussion section is some discussion of limitations, especially with experimental studies. There's always alternate approaches that pose different pros and cons. So it's always important for the authors also to discuss these limitations and for you to take them into consideration when you're thinking about what these results mean. Another point that I find to be both interesting to read and valuable is discussions of open questions and future directions. I think particularly as you're coming into a field or early career scientists, sometimes this will give you an idea for maybe future directions of the field or maybe even things for you to investigate yourself. So when reading a conclusion or discussion section, similar to what Pam discussed, it's really helpful to take notes, both to help you consolidate this information immediately, but also to refer back to it in the future rather than having to redigest the whole article. If you just list out yourself in a document or within the article, just what are these main arguments that the authors are making, and what evidence they provide in support of those arguments. And always look at these things critically. So just because the authors interpret it one way doesn't mean that's the only interpretation. So it's always important to consider alternate interpretations and other things that these results might mean, especially in light of other things you may have read. So the final section is the references section. You often don't read this outright, but as I mentioned with the intro, it's sort of a reference. So if you encounter things throughout the paper that perhaps you don't understand or you want a little bit more background on, you're going to refer to this reference section. I'm actually going to send out another poll because one thing I wanted to touch on was the use of reference managers. Please just take a second to indicate if you're familiar with any of these resources before we move on. All right. Looks like about 40% of the people are not familiar with reference managers. I'm glad I included this information, and maybe around 15% kind of evenly split between EndNote, Mendeley, and Zotero. All right, great. Pam, if you don't mind stepping along here. So there are really like a plethora of reference managers. The UCC library provides this guide that really goes in depth about the pros and cons of each. I included a screenshot from Mendeley, which is what I've been using most recently, just to highlight some of these similarities. So after you've read a paper, you have this PDF, you've gone through, taken notes as we described, all these great annotations. Maybe you've read some related papers. How do you actually organize these? These reference managers provide an interface for both storing the bibliographic information and actually interfacing with all these PDFs. The great thing about this, you can actually create folders for like a topic or a class or a project that you're working on and actually help keep all of these references organized. I think the most powerful thing about reference managers is you can actually interface them with your word processor. So this can actually save you a ton of time, both in your research and in just like your courses, if you're writing papers now. So with Mendeley, you can get this add-on. It just shows up right here under your references tab. And what it will do is launch this pop-up that interfaces with Mendeley. And you can search through your reference list and quickly add in these citations to your text. And you see it appropriately numbers them, and it'll keep updating this as you add more sources throughout your paper. And then when you're done, you can select the appropriate format or style for your bibliography. And with the click of a single button, it'll just populate it with all the resources that you utilized in this paper. So I really recommend checking out a reference manager if you aren't using one already. It'll save you a lot of time.
Cool. So just to wrap everything up, we'll just go through the main points that we've learned throughout the whole talk. So academic papers communicate original work and ideas. And literature searches leverage databases and supplementary search engines. These will help you find all the papers that you'd like to read. Abstracts or advertisements like ads. I used to decide whether or not you want to read a paper. So papers are time consuming to read. So you want to try to find things that will be relevant for the work at hand. Introductions state the significance and can guide future literature review. Methods provide a recipe to reproduce results. And results should be critically examined using multiple streams of information. Discussions, the last sections in papers are interpretive and they consider alternatives and open questions. And reference managers can be leveraged for effective literature review. So that concludes our workshop. And one last thing that I would say is that it's really important to just be patient with yourself. Like reading papers is not like a thing that we just automatically know how to do well. And it'll just take time and a lot of practice to get really fast at it. Yeah, so summary slide again. Let's see. Let's populate this. Cool. Is that Elizabeth? Is that good?
We're also happy to share these slides with you all.
Yeah. So
these are linked to all the resources.
Right. Cool. So if you guys have any more questions, you can totally feel free to put them in the chat. Oh, Mihaela, go ahead.
Hello, thank you very much for your presentation. It was great and very informative. My question is, what did you use to create this presentation? Did you use PowerPoint or something else? Because I loved the presentation. Yeah.
We just used Google Slides. So Google Drive has a whole suite of PowerPoint-like or Excel-like or Word-like applications, and we use the Google Slides.
Okay, thank you very much. Thank you.
Oh yeah, so Hira, you can unmute.
Hi, thank you for this presentation. One of the questions I had was just wondering kind of what you mentioned in terms of when you're attending lab meetings and such. Sometimes I get a little bit lost on the information. So I was kind of wondering how you go about taking notes in lab meetings and understanding where to actually delve into looking into academic papers. I don't know, it's kind of a broad question, but yeah.
Yeah, I mean, so we can both answer it because we'll probably have very different perspectives. So the first thing to know is that it'll just be different depending on what stage you're in your research career, right? So there will come a point where if you stay in the area, you're just going to know about all the things and know exactly what it is that you don't know, and that you want to look into. But I would say if you don't understand something, write down the term, or if it seems appropriate during the lab meeting, you can be like, hey, sorry, I didn't quite understand what that was about. Can you just is there a really brief explanation for it? I think I just got lost there. But if it doesn't seem appropriate, if somebody's just talking a lot, then I scramble write and word that I don't understand, or write down in my notebook things that I don't understand. And if I'm at a talk, if I'm looking at a talk or listening to a talk, I'll try to write down where I thought I got lost. And that's hard to know, but I was like, I can just be like, okay, I think I got lost after they described this thing. And, sometimes it's helpful to have like your lab mate share their slides with you if you want to like try to fill those gaps in later by yourself. So those are some of the things I do, but MJ might have a different strategy.
Yeah, I have a very similar strategy. It's like a lot of note taking. When I ever join a lab meeting, like I'll include a section of just like concepts that I know I want to review afterwards. And like my first lab meeting in a new lab, that might be like a dozen things that I write down that I have to follow up with. But week by week, that list slowly starts shrinking as you gain familiarity with these different concepts. So that's one thing I would suggest, is kind of like filling in this information on your own afterwards. If it's like a slideshow, I would recommend taking notes of exactly like what slide it was where you got lost. And so like Pam said, if it's not really a great time to interrupt, maybe you can follow up with the individual later. But I think usually it's beneficial to ask a question in the moment. I know a lot of times that can be a little bit nerve-wracking, but people really appreciate it if you ask questions. I think at your career stage, people will be impressed if you ask good questions. So yeah, I would say don't be nervous to speak up, but always take good notes so you can follow up later.
I would also say, even if you are nervous, you could just speak up also. I'm always nervous if I ask a question at a talk, like my heart is just always pumping and I'm like shaking a little bit and my voice shakes a little bit. But I mean, it's whatever. It's not like people are going to super remember afterwards that you asked like a shaky question. Yeah. So, one question, if like while we wait for people to generate their own questions, something that is like interesting to note is like, I wonder like what kinds of things have you guys in the audience like encountered that are difficult from like reading a research or academic paper? And you can either put those in the chat or like unmute and share.
For me, there tends to be like a gap in knowledge a lot of times when, for example, I'm studying different materials for use in like this device that my lab's studying, that I put together. And sometimes I just need like a textbook or like a database like value for, some property of the material, and none of them ever mention it. So I have to, I had to go find like a materials database myself that actually had some of them. But a lot of times it's just sifting through different, like just finding a resource that actually puts it all together. So because a lot of times it's, not in a way that's intuitive to like the community, or like the way it's worded in this paper. It just takes a lot of patience.
Yeah, that's a super cool point. And like I feel like that is probably what paper reading was like for like everybody in every field, like maybe 30 plus years ago, when we didn't have like ready access to the internet and like so many sources of information. So that's interestingly tough work. And there's something interesting about that, Matthew, in that like you have identified a gap in like what is available on this material, and you've actually done the work. And so like it's cool, like eventually or even now you might want to like share this kind of thing more broadly, so you can develop a blog, and like in this blog you can have a section on materials that are hard to find elsewhere, and just like have like a synthesis of what you've identified. But yeah, that is tough, and yeah, that is something that I rarely encounter, but thank you for sharing. Did you want to say anything to that, Michael?
Yeah, I think it's a very interesting point. I've come across that some too, insofar as it's an issue of communicating your science and really packaging it in a way that's translatable and transferable to other groups. So I think a lot of times that can be specific to your field. So if that's something you're currently finding to be an issue in your field, like maybe you can help to disseminate these resources either on a larger scale or just within your own lab, developing these protocols and these resources, just so those next people don't have to run into these same issues.
Yeah. So I think Angela raised a hand first, so you can unmute. Yeah, cool. Oh, nice.
Oh, hi, nice to see you guys. So I guess my question is, like when it comes to, because I have an assignment where I have to read a research article, and I have to basically go through it with my mentor, almost like present it, but like in an informal way, which is kind of like intimidating and kind of, I don't know, just a little bit overwhelming at times. So I was wondering if you guys have any tips um, in terms of just what's the word? In terms of consolidating and really summarizing the information while trying to tell a story, while presenting, the research article.
Yeah, I think some important key features of a paper to hone in on in that respect. I think first would be like, what is the gap? What is the gap in the current knowledge that this paper is trying to fill? I think that's kind of like the central crux that you might find in like the introduction, as I mentioned. And then some other key points I think are like the hypotheses, like what predictions this paper is going to make. And then another thing we mentioned was the key arguments. I think it really helps if you go through the paper, say what are the arguments and what evidence do they present in support of this. And like a lot of times you get a sense for this as you read through the paper, but actually like writing this out will really give you like something concrete to discuss with your mentor.
Yeah, I agree. I think the main thing for me is always that I have to remember to step back and just like, what is the big picture idea here? Like what is the question that these people are trying to answer, and why does it matter also? Like, why is this question important? And then, yeah, like figuring out what is the evidence presented for, like in support or against a particular like response to the question, the scientific question. But yeah, I agree that it's super tough. I definitely have a hard time with that kind of thing. It just takes a lot of like preparation, and just like really thinking a lot before sharing it. But yeah.
Come with good questions. I'm sure they're just trying to make sure that you have a good gauge for like the literature and kind of like this background research. So if there's anything you don't understand, come with those questions. I'm sure they'll help fill in the background for you.
Mm-hmm. So I think the next question is by somebody whose name I don't want to butcher. So I'm just going to say it and please correct me. Tajiri?
Yep.
Can you correct?
Said it right.
I said it right?
Yeah.
Oh.
Well, like honestly, my name has like, as long as you say the Taja part, I'm like, okay, you did it.
Okay, cool. Okay, cool. Solid.
But my question is, because my research is pretty like interdisciplinary, like it's a mix of climate science, anthropology, and journalism. And so like I'll be reading research papers, but I'm not sure who I should imitate because I'm across like multiple fields.
Oh man, that's an awesome question. I think the main guiding principle for anybody is to try to like imitate the style and structure of wherever you are going to publish in. So like different journals have different kinds of like expectations for like the kinds of articles that they want to read and like how they want to read them. And so if you're going to submit to somewhere that is more journalistic, then your focus is probably more like journalistic. So you're going to be telling a story about these like topics like the climate science and all these different things. But you just kind of have to remember that your audience is a little different and they have different expectations than the climate science journal, where they kind of want to see more technical descriptions of things, and you can bring in your perspective from like the journalistic fields. But yeah, different venues have different expectations, and yeah, I mean that's pretty cool that it's such an interdisciplinary like area. Thoughts?
Sorry, I think you broke up a little at the end there.
Oh, sorry, MJ. Yeah, I just asked if you had any thoughts on this.
No, I think you covered that one. Yeah.
Cool. Yeah, thanks for your question, to Jerry.
Thank you.
Oh yeah. So I think next is again, I'm sorry if I say your name totally poorly, but Yishun.
Oh yeah. You actually said it right. Yeah.
Oh, cool. Thank you.
Yeah. So first of all, I want to thank you for the presentation. It was so helpful. And I think we all learned a lot. And I guess my question is, because I'm doing math research. So in the math paper, it like usually requires a lot of the background knowledge. Also, they try to like prove like say 2 or 3 lemmas or propositions in one paper, but I may only like need to understand one part of it sometimes. But sometimes it confuses me because like the proof of one of the proposition will like kind of like become an obstacle when I was trying to understand another. So like for this time, like I'm not sure like where are the parts that I can take for granted and where are the parts that I really need to digest. Yeah, I think that's my question.
Yeah, that's a tough one. So full disclaimer, I mean, I don't know anything about any field other than my own. And, like I'm not even an expert in my own field. So it's kind of cool to get to hear about all of you guys' different like domains and disciplines and how papers are structured. So I think in general, you mentioned that like trying to understand a given proof is an obstacle to understanding the rest of the paper. Can you elaborate more on that? And what about it like would be an obstacle? Just because it's not relevant, you think?
Because like, for example, when they talk about the proof of some decomposition, they first talk about how it has some properties that allows it to be decomposed in this way. But like how, like why does it have that property is where it confuses. Yeah.
Yeah. So that's like that's under the category of things like, like in the example that I had of like photo tagging, it's like, I have no idea what that word is. So like, I'm not going to understand any other part of this, like, of this method section if I don't understand that verb. It's like in the title. In terms of this, like whether a given, like, you don't understand a given property, I would say that like if you think that it's core to understanding the subsequent like proofs or other things that are in the paper, if it's super important, I would take a detour and try to read about it. If you think that it's going to take way too long to read about it, I would just like mark it down in a notebook or wherever you like to take notes, and then ask about it. I would, if you have a question, don't let it go. Like you can let it go for the moment if it's not convenient to like ask somebody in that moment, but like always keep it and then ask about it later. Because like you don't have to read a paper a single time. You can read a paper multiple times. So you can always go back to it. And then after you've talked to somebody about the property that you're like unsure about, it might illuminate the rest of the paper for you again. So I think that things that are obstacles, depending on how convenient it is to answer them in the moment, like if they're super convenient, then just look it up. Just look it up and figure it out and then keep reading the paper. If it's super inconvenient in the moment, make a note of it and then ask later and then try to reread the paper after knowing what that obstacle or that challenging thing is all about. I don't know if that.
Thank you so much.
Yeah.
The only thing I would add to that would be maybe there's someone else in your lab, another student or mentor, you can ask that can quickly point you to some other resources that might be like more foundational and kind of give you that background knowledge to understand these more basic properties that are built upon it in your paper that maybe, you know, it might take you a very long search, but someone else can quickly point you to some resources.
Mm-hmm. Totally. Cool. So I think, again, I really apologize if I mess up your name. Dawe.
Yes, that's correct.
Okay.
So my question is, how do you resolve the situations which like the methodology section is lack of specificity and detail, and also like some words it just different from what has been traditionally defined?
So in terms of a method section that is underspecified, you can always feel free to email the author. So I know that that seems like kind of weird, but it's definitely like, I mean, they might not answer you, they might not respond to you, but yeah, I would just like take a chance on them actually responding to you and then you getting more clarity on how they did something. Another way to address that particular problem is, so sometimes a given paper might assume that you have knowledge of a method and then just be like, okay, and this is just like what photo tagging is. So I'm not going to talk about it here or how it's implemented. You just should read this other paper to get like oriented to that. So yeah, there might be like a previous paper that has a way more detailed methods section, which has all the detail that you are lacking.
Oh, okay. Yeah, because like last week I met the situation like I was studying some formula for autocorrelation, but this autocorrelation is completely different from the like what's on the Wikipedia.
Yeah.
This is more like a what's it called, coherence metric instead of the two something. So like this time my postdoc mentor helped me understand it, but like how do you avoid situations which words that are different from traditionally defined?
Yeah. I mean, that's hard. That's just something that will just happen. And you solved it in the best way possible, asking a postdoc mentor. Sometimes, like if a given method is mentioned that is like very specific, there will be a citation and you can look up the original paper instead of like the Wikipedia version, which might have like a similar name for something totally different. But yeah, that'll happen. That's just like a hazard of imperfect databases or like ambiguity and underspecification in terms.
Okay, thanks.
Mm-hmm. And so Luke has a question.
Yeah, I know some somewhat similar questions have been asked, but this kind of prompted mine. So sorry if it was already asked. But I know regarding words that may be confusing or unexplained. I know keywords are literally listed at the top of a lot of papers, right? You'll get a paper and it's like keywords blank blank blank. But how come those keywords are never defined? Like that's the most frustrating thing about reading papers to me is it will say like at the top keywords and then it won't actually tell me what the keywords mean and then there's never definitions for them on Google. And then I guess a follow-up is if I'm writing a paper, am I allowed to define those keywords for people or is that like is there some unspoken rule that you're not supposed to do that?
So I don't think there's. Okay, so first of all, the why are they not defined and the unspoken rule are very like have some very similar answers. Often the journal will just ask the authors of the paper once they submit their manuscript to the journal to write down keywords that are more like hashtags for people to be able to search on Google hippocampus or whatever correlation, and the paper will be tagged with that particular metadata information so that it should come up in the Google search results.
Okay, so they're not like terms to know, they're like labels.
Yeah, they're terms that like are meant for people to like identify relevance, whether that's a metadata feature for search engines or whether that's like for people who are just browsing through papers trying to figure out what to read. They're like, oh, this is about the hippocampus and it's about like spike timing, or it's about like something else. I don't know. So that's not something that most authors, I think, even think about those keywords. They're just like, okay, this paper is about hippocampus X, Y, Z, whatever, and then submit. I think also, yeah, like papers have very limited space constraints. So you might have like some papers that are like 15,000 words that you're allowed. Other papers are like, this paper can be no more than 6,000 words. We have like very small print or whatever, or small page limit for this paper. So authors are not going to add information into a paper that they think that you can get elsewhere. So, they're not gonna, like, if they have a really tight space constraint, they might not like define super in detail all of these terms because they know that you can find another paper that has a super specific description about that term because that's what the paper is about. So,
gotcha. Okay.
Yeah.
That makes a lot of sense. Thank you.
Totally. Okay, so it is 5:01. I think we should probably wrap up. Thank you guys so much for how engaged you are. I was really surprised at how like awesome you guys are about participating. So thank you so much. And feel free to like email us with questions. We can give Sophia and other program coordinators our emails to pass on to you guys if you want to connect in some way. So yeah. Have a good.
Thanks so much for your attention, everyone.
Bye, guys. And good luck with all your research.